Hello, my name is Tyler Bell. I'm a postdoctoral scholar at the University of California, San Diego, and my advisors are Carol Franz and Bill Creeman in the Department of Psychiatry. And today I'm going to be talking about how chronic pain throughout midlife affects the locus coeruleus integrity as older adults enter older adulthood right at the age of around 68. And I'm going to take a moment and share the slides. All right, here we go. All right, so why I'm interested in the locus coeruleus in my first year as a postdoc is because the previous research that I've been doing has been looking at how pain affects cognitive decline and impairment risk. And it's shown what some other people have shown, which is that throughout life, the persistence of pain increases your risk of cognitive impairment, dementia, as well as just poor performance over time in various domains. But the biological reasons in between pain and cognition and what would lead to impairment remain a little bit unknown. And so I want to focus on the locus coeruleus through some guidance provided through my mentors, and particularly because the locus coeruleus is a very vital part of the brain that is the host of what produces most of the norepinephrine in the brain. It's located deep, if you look under the methods section where that blue square is, very deep within the dorsal pons of the brain. It's only about 33 cubic millimeters big, but despite that, it has very wide projections throughout the brain, as you can see in the red lines in this method section. And it also produces almost all of the norepinephrine that is in our brain. And we know that norepinephrine is important in the brain because it's important for sensation, vigilance, as well as cognition and attention and wakefulness. It's also been implicated in some clinical things like sleep disorders and major depression. But we haven't been really able to study it in vivo until just recently with new technology that I'll get into in a second. So the locus coeruleus has been implicated in cognition, specifically lower integrity or lower volumes post-mortem have been related to poor cognition and increased dementia risk, especially within the rostral region. And that makes sense because that rostral region projects into vital cognitive neural substrates, including the hippocampus and the frontal lobes. And then when we look at the locus coeruleus, we also know that it also affects pain perception. And this has been shown in a lot of animals, particularly rat models. The caudal region in particular has a lot of projections towards the dorsal horn, and that's thought to be a key modulator of what will enact descending inhibition, pain inhibition, or facilitate pain detection into the central nervous system. And so the locus coeruleus, because it has both a hand in cognition as well as other different things important with perception and everyday functioning, but also has a big hand in pain perception, I think that this may be a key biological link in between how pain affects these various outcomes. And so the purpose of this study was to examine how chronic pain presented throughout midlife affects the locus coeruleus as individuals jump into older adulthood right at the age around 68. And the purpose of this study is to examine this association with the key hypothesis that chronic pain across midlife will be associated with a lower locus coeruleus right at that entering older age benchmark. The participants derived from the Vietnam era twin study of aging. This is a 100% male sample, twin sample, and this was required to do twin analyses. So just keep that in mind that this may not fully be generalizable to a lot of the female cohorts that we also care about and look at. Chronic pain was measured throughout VETSA 1 and 3 through the SF-36 Quality of Life Scale. There's a bodily pain scale that asks individuals 2 questions about pain, pain severity, and pain interference. I'm looking at pain severity in particular, which asks people in the last 4 weeks, how much pain severity have you had? And individuals were marked as having chronic pain in our study if they presented moderate to very severe pain for 2 waves or more. And those aren't years, those are, the waves actually are conducted nearly every other 5 years. And so across 12 years, if individuals reported moderate to very severe pain over 2 or more waves, which is a substantial amount of time, those individuals were denoted as having chronic pain. And just moving up for a second, individuals were screened um, for various components before they got into the MRI. So there were some things that they were screened out for, such as epilepsy and some other conditions. And only 485 individuals at the third wave did the MRI subpart of the study and provided LC images using a new technique that I'm about to discuss in a second. And just keep that in mind. The cohort is a lot bigger than this 485, but this is our MRI sample within VETSA. For the LC imaging, we use GE3T Discover scanners with T1 weighted images. And the regions of interest that we looked at were the rostral and caudal locus coeruleus, or LC regions. And you can see in the two crosshairs in this dark image, how we got the volume was we found a detection point of where we thought the middle of the LC was, and then we do detection of the left and the right LC using these cross marks that a lot of imagers in our study placed on the map. And using those crosshairs, we were able to collect volumes of the left and the right LC. And in particular, what we were interested in for this study was to detect the level of neuromelanin signals to get at this volume. In particularly, let me back up a little bit. The locus coeruleus, this imaging method sliced the LC from the middle in the left and right segments. And then we were able to calculate the total signal score using the neuromelanin. So neuromelanin is this byproduct of the production of norepinephrine. And because the LC should be producing most of the norepinephrine, we are able to use signal detection to determine how much neuron-producing cells are there within this region compared to some reference region. And for this study, we use the pontine tegmentum as a reference region. So any signal detection there should mostly be noise, and we're able to control for the signal that is relevant to neuromelanin and what's just noise using a contrast to noise ratio, as you see here as the LC-CNR. So taking this metric, this will tell us how much neuromelanin is being detected in the LC regions, which should be a proxy for the amount of LC cells or volume, as well as the health of the LC. So higher values are going to indicate better LC integrity. We convert these values into z-scores just for easier interpretability, using the descriptives for the rostral LC-CNR and the caudal LC-CNR at this third wave. Important to know about the analysis I'm about to present is that we eventually control for a lot of comorbidities, depressive symptoms, age, and opioid use that could also be affecting the LC. And just to let you know, we do eventually control for those in this study. So looking at the results, what you can see in this graph and in this table is that individuals with chronic pain had substantially lower locus coeruleus contrast to noise ratios in the rostral and caudal regions. And what you also see is that individuals were more different on the caudal region, which makes sense because we think that the caudal region, because it projects to the dorsal horn, is being more involved in pain processing than the rostral region. But we still see it for this rostral region, this area important for cognition and wakefulness. And these results remained even after we controlled for a vast host of these covariates that may be affecting the LC, such as age, depressive symptoms, and comorbidities, as well as opiate use. And none of the other covariates did really contribute to the model, and the main driver was this chronic pain. So in conclusion, chronic pain in midlife really does seem to be related to lower LC integrity in older age. But whether or not this is a causal effect, more of an individual difference, so maybe some individuals develop chronic pain because they have lower LC integrities for some reason, remains to be tested and explored. Neuromelanin imaging is a new technology that has come out in the last couple of years. So I expect that, such as in VETSA, we're going to see some longitudinal data that we can start looking at, or longitudinal associations. And even in twin modeling, we can do some causal analyses if we get some more longitudinal data. It's important to note that the associations were not explained by depressive symptoms, comorbidities, or opioid usage. And it's even important to note that opioid use actually kind of trended and nearly significant to better LC volume. So maybe pain management is important to some degree to keep these LCs healthy. Pain's effects were shown for both the rostral and caudal regions. So unlike the animal research, we don't know whether or not there really are specific effects, but both of these regions are associated with a host of different clinical symptoms that may be involved, such as wakefulness, sensation, and cognition. We know that fatigue and depression are major issues in these samples of people with chronic pain in particular, and maybe the LC is something at blame. We also know some pharmacological ways to improve norepinephrine in the brain, such as SSNRIs, have been effective in reducing pain, and maybe this is why. But again, that's all very speculative, and we have a lot more research to do. But as a first step, the LC seems to present an important biological link between chronic pain and cognition, and various different outcomes that we can look at, like sleep and attention, for future research, that we should start to explore, especially as we can now look at these deep brainstem regions with neuromelanin imaging. I just want to say thank you to my lab, who have been very supportive in helping me learn some new ways of doing analyses and some new neuroimaging methods. And if you have questions, feel free to email me. My email is below here at trbell@health.ucsd.edu, or you can, throw a comment at me at Tyler Bell PhD on Twitter. You can also feel free to email me if you want to get in discussion and talk to my mentors, Bill Creeman and Carol Franz, if you're interested in looking at some of this data yourself, or just getting involved in some collaborations. And I thank you for your time, and I would love your feedback. Thank you.